Good morning, everyone. Welcome to the Arthritis web seminar series. It's our lecture number 30 today on cellular senescence and arthritis. Our guest speaker today is Dr. Johnny Huard from Colorado State University, Vail, Colorado. Before we start the introductions, just a reminder to the audience, please post your questions on the question answer tab on Zoom. Over to you, Rob.
Thank you, Mohit. It's a great pleasure to welcome Johnny Huard to our web seminar series. Johnny is a Canadian by birth and by early training. He's currently the Chief Scientific Officer and the Director of Center for Regenerative Sports Medicine at the Steadman Philippon Research Institute in Vail, Colorado. This is a lovely background picture from his window, his lab, I think. That's a wonderful place to be living, I'm sure. Johnny's had a very distinguished career, extraordinary contributions in gene therapy, tissue engineering, regenerative medicine. Prior to his time in Vail, he was the professor distinguished chair in orthopedic research in the Department of Orthopedic Surgery at the University of Texas Health Science Center in Houston, and also had a strong affiliation with the regenerative program in Pittsburgh. So Johnny's made a singular series of contributions to the field of regenerative medicine as it relates to arthritis and bone disease and also stress incontinence. And it's a real pleasure having him join us today. Welcome, Johnny. Thank you.
Thank you. So this is a real pleasure to be a speaker to your seminar series here. And again, thank you for the introduction. So what I'm going to try to do here is to give you a couple of, stories that I like, to tell about, how the science sometimes start at the bench and end up to the bedside. And of course, I'm going to be talking about cellular senescence and osteoarthritis. So I cannot advance my slide. Hold on. Okay. So here is really, first I would like to acknowledge all the funding support from the NIH, as you can see here, and the Department of Defense. And I just would like to say that all the people here that you see in the bottom of the slide are, a grateful donor that came to the Steadman Clinic in Vail and get treated and gave back to the community and gave back to us to do some research. So we get a lot of philanthropic gift from those people that really support the research. So who are we? So we are a team, as you can see here, I'm the chief scientific officer there, but Mark Philippon, an older Canadian, is a managing partner of the Steadman Clinic, and the Steadman Philippon Research Institute is one of the premier, orthopedic surgeons that deal with the hip. And everything was started with Dr. Steadman, in the mid 80s. And here is now, the list of orthopedic surgeons in sports medicine that work with us, as you can see here. And, now we have a site, not only in Vail, but we also have a site in Aspen. So we do all the orthopedic surgery care in Aspen as well. And what you see here, this is all the clinical fellows that come from United States or abroad that come here to work with us, and they serve as a bridge between the scientist and the surgeon, because they can go in surgery with the surgeon and they do science with us. So I really believe that they are a very important component to our research program. So what do we do? So everything we try to do is to improve orthopedic care. And of course, we have 4 distinct departments. The regenerative medicine is a department that I'm leading, and again, we do a lot of biological approach to improve tissue repair, as you can see here, and I'm going to be talking about this today. But we have an imaging, an MRI, department that assess, tissue structures, monitor healing, and degeneration. We have a biomotion, a full body and a whole body performance biomotion that work also, in conjunction with us. And of course, we have a biomechanics and robotics, try to understand the anatomy, evaluating, device and procedures. And finally, that's something that Dr. Steadman has been very famous, for, is to set up a clinical outcome. We have patient-focused outcome assessment. We have almost a lot of people that has been, treated here. We still follow them. We still follow, they come regularly in the clinic and to be evaluated, because a lot of our treatment here is to try to delay osteoarthritis, and sometimes it happens 10 years down the road after surgery. So I'm going to be talking about the regenerative medicine program, that I built and I set up, in Vail, Colorado, starting in 2015. So here is the regenerative medicine program, where everything we do here is to try to develop biological approach to improve tissue repair after injury, disease, and also aging. And you will see today that we're very excited about aging research, and we have been really, trying to apply this to the clinical arena. So the first pillar of research is really biologics. So we're using a lot of adult stem cells. Of course, I've been known to work with muscle stem cell for a long time, but now we have a variety of stem cells we can use from. And also orthobiologics, platelet-rich plasma, bone marrow concentrate, or treatment is being used in a clinic every day. So we're trying to understand how they work, and that's going to be one of the focus of my talk today. We are doing a lot of regenerative medicine, and as you can see here, we're doing a lot of gene therapy, CRISPR-Cas9 gene editing, but we're using a variety of, scaffold or exosome, vesicle, quadrate, microspheres, nanofibers to try to deliver, genes and proteins to the site of the injuries. The third pillar is therapeutics. What we try to do here, knowing that, if you discover a new stem cells or new drugs in your lab, on average, it takes 17 years before this thing, spill in the clinical arena. So what we decide to do here is we try to look at FDA-approved drugs that they are being already approved for one indication that we use as an off-label to orthopedics. And I'm going to give you a good example of that, with Cosar or losartan, a blood pressure medication that now we're using for osteoarthritis. And finally, animal modeling. So everything that we do need to be tested in small animal model and sometimes in large animal model. And we do this at the Colorado State University in Fort Collins here. So this is really the four pillar of research that we have. But today I'm going to try to see, to show you that we're trying to improve orthobiologics treatment, and we're trying to improve their efficacy by using off-label drugs or natural supplement. So here is really, how we're set up. So the research is being done at the Steadman Philippon Research Institute, which is a nonprofit. And as you can see there, we have, 6 NIH grants that try to develop the biology of the future. So this is all mechanistic studies being done in animal model at Colorado State University. But we also have, a couple of clinical trials here that you can see. We have 5 of them that now is the biology has become to a point that, it's close to the clinic. And now what we're doing here, we're testing those, biologics in double-blind clinical trials with a placebo group. And if it works there, then it's spilled to the clinical, application that we do at the Steadman Clinic. So clearly, this is the pathway that we're bringing science to the clinical arena. So I want to talk about osteoarthritis, and I know that you probably know that this is, characterized by a destruction of joint cartilage, leading to pain and loss of function. And this is really a debilitating disease because it affects a lot of people. The older we get, we all end up to have osteoarthritis. And also at the sports medicine clinic here in Vail, everything that we do is somehow linked to osteoarthritis. But you also know that post-traumatic osteoarthritis is even more, difficult to deal with because it affects, younger people that have an injuries and after this end up, to have, osteoarthritis. So clearly, this is really the target that we have here. Can we develop approach to delay osteoarthritis? So the first project I'm going to be talking about is the use of platelet-rich plasma and bone marrow concentrate to treat, ACL, ACL reconstruction after ACL injuries. So first, that 50 to 90% of ACL injury progress to post-traumatic osteoarthritis. Yes, we're successful to fix the ACL, but 10 years down the road, those patients develop osteoarthritis. And some study demonstrated that ACL reconstruction not only cannot fully prevent the development of OA, but in certain occasions, ACL reconstruction may be associated with a high prevalence of OA. So this is why, we're trying to work with ACL reconstruction to try to use biologics, to try to develop approach to delay OA down the road. So here, this is the orthobiologic market, and again, this is a pretty, popular field right now. But the problem with orthobiologics is some people like it, some people hate it. And again, the main product in orthobiologics that has been used so far has been platelet-rich plasma and bone marrow aspirate. And again, I was also part of a AAOS, American Academy of Orthopedic Surgery, a consensus, meeting, where what we end up to say at the end of this meeting was that we need to put evidence behind those products, because right now we don't have any evidence. So again, just to give you an example, orthobiologic for ACL reconstruction, there is a positive clinical trials investigating the role of stem cells in promoting ACL healing. The role of PRP is still controversial. Some people say it works. Some people say, well, it doesn't work. I don't think you're going to hurt your patient, but I don't think sometimes you have no effect whatsoever. And the cost of those technologies is not covered by the healthcare. So sometimes what we want to do is to try to understand how it works. So the way we do this, here in Vail, Colorado, so every time a surgeon upstairs do a PRP or a BMAC, a fraction of this PRP and BMAC come in the lab, and I'm looking at a lot of different factors within PRP, like inflammatory cytokine, angiogenic, factors, fibrotic cytokine, and I put this in a bank. The same thing with BMAC. I'm looking at different population of cells like endothelial cells, pericyte, mesenchymal stem cells, hematopoietic stem cells. I'm giving a ratio, and I put this in my bank. And the idea behind this is, after, we have done, a lot of that. So I go and see the patient, the surgeon, and I tell them, can you tell me what, which patient benefit from your PRP? And if they tell me patient 10, 20, and 30, I go back in my bank and try to find what those orthobiologics product has in common. And the goal is to get a signature. Again here, BMC sample, we're quantifying cells, within the BMC sample. The PRP sample, we're quantifying, growth factors. And I'm not looking at single growth factors. I may look at 5 clusters of factors that affect angiogenesis, factors that affect fibrosis. And I put this, in the bank, as I said. So just to give you an example how that works here. So I think you can probably see in this slide here, if you want to reduce inflammation, then interleukin-1 receptor antagonist protein is one of them that you may want to look at. And if we look at the amount of interleukin-1 receptor antagonist protein in those different orthobiologics products, it turns out that BMC, bone marrow concentrated, contain the most. So if you want to reduce inflammation, maybe BMC should be your target. Okay. If I want to look at angiogenesis now, again, for muscle repair, for example, angiogenesis is very important. So we look at angiogenic factor here, and we find that the leukocyte-rich PRP has more angiogenic factors. So maybe if you want to promote angiogenesis, then maybe, leukocyte-rich PRP can be your target. If you want to reduce fibrosis, then we're looking at TGF-beta1 because we know it drives fibrosis. So we look to all the different products here. And it turns out that the BMA, bone marrow aspirate, is the one that contains the less, TGF-beta1. So clearly here is really to try to get some indication, what are those different products contain, and maybe they can be selected for a particular application. So now, we would like to develop a signature profile of PRP and BMAC, to try to target some injuries. It can be muscle injuries or cartilage or ligament. And here is a first clinical trials that we're doing here. So we're doing ACL reconstruction, in 60 patients here, 20 got ACL reconstruction plus we had PRP, 20 we get ACL reconstruction and we had BMAC, and 20 we have ACL reconstruction that we use as a control. And again, we look at biomotion and imaging to monitor the healing process. And the goal is to see and provide evidence if PRP or BMAC work, and also identify the factors within PRP or BMAC that may work to improve ACL reconstruction healing after surgery. A second project I want to talk about is this project here called about macrofractures or biologically regulated marrow stimulation. And we find that blocking fibrosis in the context of cartilage is very important. And here I'm going to share with you the study that we're doing here. First, macrofracture was developed, right here in Vail with Dr. Steadman. And as you can see here, He was using a pick basically to make hole in the subchondral bone under the cartilage lesion. And the goal there was to have some bleeding from the bone marrow bleeding in the joint to improve cartilage repair. So that was very, easy to do. The problem with that, though, is the cartilage that you're making is not very good hyaline cartilage, is mainly fibrocartilage here. And me, I've been working for almost 25 years to try to prevent, fibrosis and skeletal muscle. And we have known for many years that TGF-beta1 is the major driver of fibrosis in muscle. And what we have found in Pittsburgh is that if you block TGF-beta1 with TGF-beta1 inhibitor, can be Decarin, Suramin, gamma interferon, relaxin, and also losartan now, if you block TGF-beta1 to improve muscle healing after injuries. But nobody attempt to block TGF-beta1 for cartilage repair because TGF-beta1 is the main cytokine that turns stem cells into chondrocyte. So what we decide to do is we decide to ask, is fibrocartilage also fibrosis developing in cartilage? And if we block TGF-beta1, can we improve, cartilage repair by eliminating fibrosis? And here's a study that we have done. So we basically do macrofractures in a rabbit studies, and some animals get CoSAR in their drinking water and some do not. So here's the result. I mean, you can see that, if I look at the bone marrow stimulation, so you get a pretty good repair process. But if you do the macrofractures now, and if you add losartan to that treatment, you're making very good hyaline cartilage here, as you can see here. And you have a very good distribution of collagen type 2. And on the scaling to grade our cartilage, it turns out that adding losartan significantly improves articular cartilage here. But what we did here is we monitor the amount of fibrocartilage. And we're using, a staining, Aerovici staining, to really evaluate the type of cartilage we're making. And as you can see here, if you use losartan with microfractures, you make a very good hyaline cartilage, almost textbook. And when we monitor the amount of fibrocartilage, when you had losartan, you reduce the amount of fibrocartilage to 10%. So that means that the cartilage we're making is almost 90% hyaline cartilage. And we believe this is very important for the long-lasting cartilage that you're going to develop, because fibrocartilage will not last long. And we believe hyaline cartilage will last longer. So losartan block TGF-beta1 and will likely reduce fibrocartilage in OA patients. So now here is a good example, something that started in the lab, and we did some rapid studies. And in 2019, not only we got the ON Ortho Regeneration Award from the ORS, but we also got the Cabo Memorial Award at the American Academy of Sports Medicine. And this is a perfect example here of something that started in the lab that ended up in the clinical arena. At the same time that we were doing those studies, Mark Philippon started to use losartan in his clinical patient. And as you can see here, when he has, patient, that come in, 4%, come back because they need a revision because the surgery was not, optimal for them. But if he had losartan to those patients, he cut his revision case from 4 to 1.9%, which is a big deal. But when you do revision case, so that means case that started somewhere else in the United States, they went to see a surgeon, it doesn't work, they come in Vail, they are complicated case. In those cases, 15% of them as revision because they are very complicated case. But adding, losartan cut this to 2% revision case. So clearly, it shows that, losartan, a simple drug that can be added to after surgery to your patient, can cut your revision case. And because of that, of course, we have a second clinical trials right now where we do, a double-blind study where some patients get losartan after microfractures in the hip and some patients do not get losartan. But I have to tell you here, when we start these trials in 2019, Mark was still collecting data from his patient. And now losartan works so well in his clinic that it has become a standard of care. So for us, it became, a real problem because, Mark didn't want to put any patient on placebo because losartan was so good. And because of that, now we decide to do the study where we're going to take all these patients that get treated between 2011 to 2015, where he was not using losartan, and he was compare the success rate of, surgery with patient, after 2016, where he used losartan all the time. So that really indicates that losartan seems to work here, and it can be, easy to apply and very beneficial for the patient here. So although we're conducting clinical trials, the science at the bench do not stop. And here's a good example of that. So we have two papers back to back, in the American Journal of Sports Medicine in June and July of 2021. This paper here show that, if you do an intra-articular injection of losartan, then, you can also improve, cartilage repair. Why we want to do an intra-articular losartan is because losartan is a blood pressure medication, and maybe giving it orally, sometimes you may have a risk to reduce the blood pressure of your patient. So therefore, the intra-articular, it's maybe most important. The problem, though, is I want to tell you is the dose is very important here, because if you inject too high of a dose into the knee joint, you may have a detrimental effect. And the other study here, we're using another drug called, Avastin, a drug that blocks, angiogenesis, a drug that has been used for the cancer treatment for many years. And now we find that if you add Avastin to the microfracture, you improve the benefit of the microfractures by blocking angiogenesis and by making, hyaline cartilage. And here, those people here on the bottom of the slide here, this is all clinical fellow, either international or US fellow that come here and participate to that research. Another project we're doing now is losartan also beneficial to other joint. Now we have very good, result, for the knee and the hip. But how about the shoulder? When you do a rotator cuff, and here, this is an example of a study we started to do in animal model, that we found that losartan can also improve rotator cuff healing after, microfractures. And here, just to tell you, a few weeks ago, we just got an award from the Germany Society to support that work. And finally, we also try to use losartan for the ankle injuries to try to, when they do microfracture there as well. And the goal here is to use, losartan to block fibrosis. Again, guy, now I'm being invited around the world to talk about losartan or COSAR. And I said, wait a minute, I want to talk about something more exciting than COSAR. But what I realize here in science, in orthopedics, is sometimes the easy thing, it's what make a big difference in clinic, because losartan here, this is not a new stem cell, this is not a new scaffold that need years of regulation by the FDA. Just adding a drug to your surgery here, can make a difference. And you use the drug as an off-label. So that's very important. That's a lesson I learned from that. So here is a study that we're very excited about, because we found that with osteoarthritis, you have an age-related decline in the chondrocyte ability to repair articular cartilage. So we know that. But what we start to realize here is when the chondrocyte, reduce their ability to repair their cartilage because, they lose their chondrogenic abilities or the regenerative potential, we find that you have an increase of cellular senescence into the joint fluid, into the joint cartilage. And again, that was published, by Jody Buckwalter in 2001, where he showed clearly that osteoarthritis is related to cellular senescence somehow. The problem is he came to give us a grand round, probably a year ago here in Vail, and asked him why you didn't push this science. He said, well, back then we didn't have any drug to eliminate senescent cells. So for me, I didn't have an intervention that I can do. It turns out that 20 years later now, we have an intervention, because now we know that cellular senescence it's a huge, investigation area, in science now, because those senescent cells, they stop dividing, they stop proliferating, they don't become cancer cells, but they secrete a lot of factors that drive inflammation. And, and as you know, inflammation is driving aging-related disease. We know cancer, neurodegeneration, are linked directly to inflammation. And now we know that the senescent cells play a key role there in the development of those diseases. But we also know now that senescent cells play a key role in osteoarthritis. And I'm going to share with you, some research on this. But the bottom line is why we're so excited about this now and why it's so popular is because we have an arsenal of drugs and supplements now that eliminate those cells specifically. So they basically target those senescent cells and induce apoptosis to senescent cells. And some of those drugs are not even drugs. They're natural supplements. They're plant derivative that you can use, you can buy from GMC or on Amazon and so forth. So again, here, this is the work we're doing with Jim Kirkland at Mayo Clinic. Here is really, we find that senescent cells are involved in all those diseases, as you can see here. The red here is a disease that I've been working on in my lab here in Vail, Colorado. But the bottom line here, this is all disease where senescent cells seems to play a role and where, senolytic drugs can probably have an effect on to delay the disease progression. So here's a great paper from Jim Kirkland. What he found here, he found that if you, concentrated senescent cells from a whole mice, and after this, you inject this into the knee joint of a young mice, then you develop osteoarthritis, showing that those cells drive the disease process. And as you know, for many years, the way we treat osteoarthritis is we're not treating the disease, we treat the symptoms, so we eliminate, we try to reduce inflammation. But here, if we eliminated those cells, we think we will be very, it will be a very important, intervention because it would prevent inflammation to develop further down the road as well. So here we use, an animal model, a progeria animal model that you can see here that, basically develop osteoarthritis like you and me. And those mice, are missing lamina A. They are like the AGPS, progeria syndrome in patient. But those mice, are short-lived. They live for up to 6 months. But what I'm so excited about those mice is they develop osteoarthritis like you and me. A lot of osteoarthritis animal model that we use, we have to damage the meniscus, we have to cut the ligament. But here, those mice develop spontaneously osteoarthritis without having to injure them. And what we have found also in those mice that you have a lot of senescent cells in various, area. We have some senescent cells in their muscle, in their peripheral blood, in their bone marrow. And you can see that, not only we see senescent cells, but we see also, inflammatory mediators that they secrete, and so forth. So can we delay osteoarthritis in this progeria animal model by eliminating senescent cells? And this is what we have done. And here's very simple. We put those mice, some mice got the, the senolytic agent, in their drinking water, and some do not. And here, this is a progeria mice that do not get the senolytic agent in their drinking water. So you see degeneration of cartilage, you see lots of proteoglycan, and you see a lot of cells here that express P16, which is a marker for senescent cell. This is not the only marker for senescent cell, but this is one of them. But the most amazing thing is when you treat those animals with senolytic agent here, you preserve articular cartilage, you preserve proteoglycan, and you eliminate a lot of senescent cells in the articular cartilage area. So again, here, this is not a way to regenerate cartilage. This is a way to prevent articular cartilage degeneration. And the most exciting thing about this is now here, we didn't even use drug, we use natural supplement that can be used to delay osteoarthritis. And because of that, then we get a clear path with the FDA and we get approved to do these clinical trials here, where we have 72 patients with moderate OA. Moderate is very important here because we don't want patients too far, into the OA degeneration and they need a knee or hip next week. We need patients that are middle OA, so that means that they have pain, but they're not ready for a joint replacement. 36 will be on fisetin, which is the natural supplement that eliminates senescent cells, and the auto 36 is placebo. And again, as you can see that we monitor the healing at 4, 8, and 12 months, and the goal is to delay OA progression. Also, the goal is to delay the first joint replacement as long as possible, because when you get the joint replacement, sometime, you will have to have one 12 to 15 years later. So we hope if we can delay this, then the joint replacement you're going to have will be the last one you're going to have. So now I told you that, yes, we have senescent cells, and yes, we know now that, they are involved in disease progression, and yes, we have drug to eliminate them. But what we didn't have over the last 2 or 3 years is, do we have a way to detect senescent cells? So, it's like cholesterol. I mean, we're not going to give statin to people that we don't know if they are not making high cholesterol. So I think it's very important to know, how many senescent cells you have in your body. So here, that brings me to a test that we developed in the lab here. So we have, 250 participants that participate to this work. And we use, basically the C12 FTG, which is a senescent cell detection. because it detects beta-galactosidase. And on this graph here, we have, this is the distribution of our patient. The blue is men and the pink is woman, of course. And what you see in the tip, here is patient that start to take fisetin, either for women and men. And we look at senescent cells as well. So here's a result that I can share with you. So if you look at, a T cell, T senescent cells, young adult, old adult, we can clearly see that, in whole adult, you have more senescent cells, T cells. But if I look at PBMC here, peripheral blood mononucleated cells, we can see also an increase, when you get older, then we have more peripheral blood mononucleated cells that become senescent than when you are younger. So we're still working. So the bottom line is, older we get, more senescent cells we have. And also what we do here is we correlate the amount of senescent cells with biomarkers, OA biomarkers. And as you can see here, we have a positive correlation between the number of bright senescent cells and those biomarkers here. And we also look at the biomarkers and aging as well, and we can see that we have a positive correlation with OA markers. So we knew that OA markers, biomarkers, increase in our blood as we age. But now we know that more senescent cells you have in your body, more biomarkers, OA biomarkers, you're gonna have in your blood as well. But here's the good news. So now this is a patient that get tested for us at baseline. But if you take fisetin, 100 milligram per day for 30 days, we seem to reduce the number of senescent cells that you have in your blood. It's very efficient. And here's another way to look at this. So the red bars are people that came at baseline here, and the black bar are people that take fisetin for 1 month. So you can clearly see that we can reduce the number of senescent cells in their blood by just taking fisetin, a natural supplement, 100 milligram per day. And that was very interesting, and we're following this work. Not only we reduce the number of senescent cells by treating with fisetin, but we also reduce the number of factors that those senescent cells are secreting, like inflammatory mediator, TGF-beta1, and so forth. So clearly, fisetin reduces senescent cells, but also reduce the SASP that those senescent cells will secrete in your blood. So here, we wanted to know, how fast fisetin works. So we did this work in culture. We take the blood and we put the senolytic agent or the fisetin directly in the blood, and we look at the number of senescent cells. So you can see at time 0 here, we have a large amount of senescent cells, but after 1 and 4 hours of fisetin, you can see a drastic reduction in the number of those bright senescent cells here. The senescent cells here, the bright senescent cells are shown in green. So clearly it worked very fast. The second thing that we did here, we found that when you treat with fisetin, not only you reduce the number of senescent cells here, but you also increase the number of cell death. So that means, fisetin kills specifically those senescent cells here and make them, promote apoptosis to those senescent cells, so they die. So when those senescent cells decrease in your blood, the number of cell death increase as well. The number of senescent cells death increase. So again, this is very exciting because when fisetin reduce your senescent cells, at the same time it increases the number of dead cells because we eliminate those bad senescent cells. So now how that compare with other drugs. So we have fisetin now, quercetin and dasatinib and metformin, and you can see fisetin is very fast to induce cell death to those senescent cells. Metformin and dasatinib, quercetin and dasatinib here, they're not as fast, but around 18 hours you get what you're getting mainly with fisetin. And with metformin and other senolytic agent here, it takes 18 hours to eliminate those senescent cells. So those cells has a different kinetics, how they work. And this is why we like fisetin, because it's very fast. And again, you increase the number of cell death there. So what we have now, we have a program that we optimize longevity through precision medicine, where we're going for full blood work because we want to know, a lot of things in your blood that nobody have looked at, toxicity, to metals, and we're looking at leaky gut and all sort of things. And what we do know now is when you come to be tested here at baseline, you know, and if we give you fisetin, you know, we always reduce the number of your senescent cells, as you can see here. And at the same time, when you come at baseline and we treat you with, you know, fisetin, we also reduce the number of P16 expression, which is a markers of senescent cells. And again, when you come at baseline and we test and you take fisetin for 1 month, we also reduce, you know, OA biomarkers like MMP1 and MMP3. So clearly, you know, now we have 250 patients that we have been doing this on. So now, you know, we detect two populations of senescent cells. We detect the very bright one. They're very bright here by on the FACS sorting. And we have those ones that are not so bright. So we have the normal cells, the one that are moderate, you know, fluorescence, or, you know, they are, you know, dim. We call them dim cells. And we have, you know, the very bright one. So when you take those three fractions and you look at the expression of P16, it turns out that the high, you know, bright senescent cells express more P16, they express more P21, and of course, they express more interleukin-1 beta. So we think that those bright cells are the worst senescent cells you have, and we believe that those cells are the one we need to eliminate. So now what we want to do is we want to take the high senescent cells, the mid senescent cells, or the dim, and we want to inject this into the knee joint, and we want to see which one will develop osteoarthritis faster. We believe that the bright will be the one that will drive osteoarthritis, but we're still doing the work now. This is part of the work we want to do, you know, here. So another thing that, you know, you can ask is senescent cells only present in your peripheral blood? And the answer is no, because we find senescent cells present also in your bone marrow here. And we know that, you know, older you get, more senescent cells you get in your bone marrow as well. But here, this patient, you know, it was 57. It had a very high senescent cells, and we didn't know, you know, why. It was relatively young and high senescent cells, and it turns out that this patient has rheumatoid arthritis and gout. So I believe that the number of cells that you have in your bone marrow, senescent cells, I mean, it may be indicative of a disease somehow. Synovial fluid, the same thing. We aspirate synovial fluid now, and we can see some senescent cells in your joint fluid as well. So right now is, you know, we have senescent cells in peripheral blood, bone marrow, and joint fluid. But the question is, you know, are they the same cells? Are they different cells? And I think they are different. And we're doing this work now where we take synovial fluid, you know, senescent cells, or senescent cell from bone marrow, senescent cell from peripheral blood. And we're going to do a lot of analysis like multiplex biomarkers, analysis, and so forth. We're going to do, of course, you know, FACS sorting and everything. But one thing we want to do is we want to do single-cell RNA-seq on this. And that's the work, you know, we would like to do in collaboration with the group here, you know, in Toronto, is because they have this technology that we can do. And of course, you know, we can do data analysis, you know, and computational modeling, because we would like to know, you know, the population, can we identify a unique population? I can tell you so far that the senescent cells, you know, in your peripheral blood are mainly monocytes. So we know that. We have been collaborating with the Buck Institute, but more work needs to be done. And now I'm going to finish with the final clinical trials that we have been doing is now, you know, can you use senolytic agent and antifibrotic agent to improve the benefit of bone marrow stem cells? So now we do bone marrow concentrated stem cells. But in these trials here, 1 month before, we start to treat the patient with fisetin to reduce inflammation before it comes to surgery. And when we did a bone marrow, you know, injection here, then, you know, for the next month, now we give them losartan to reduce scar tissue. So we reduce inflammation before, reduce scar tissues after. So we're doing a phase 1/2 randomized clinical trials where we want to detect whether fisetin and losartan will improve the benefit of bone marrow aspirate for knee OA. So again, what's the rationale behind this? We know that when bone marrow, we know that older you get, more senescent cells you get in your bone marrow. Not only you get more senescent cells in your bone marrow when you age, but you also get more SASP factors that are secreting in your bone marrow. So that really indicates that maybe senescent cell can be a benefit to combine with the bone marrow, because, you know, in older patients, bone marrow contains senescent cells. Is it working? Well, we have done the studies in vitro here where we take, you know, bone marrow, you know, MSCs, and now some of them, we treat them with fisetin. And you can clearly see here, when we treat with fisetin, we decrease, you know, the amount of, you know, senescent cells in your bone marrow by 22.5%. So clearly, it indicates that yes, maybe fisetin can be used to reduce bone marrow, to reduce senescent cells in your bone marrow to improve the benefit of your bone marrow. So senolytic agent can eliminate senescent cells and likely reduce, SAS factors in your bone marrow and should improve the benefit of bone marrow for OA patients. So here is the paradigm. So we think that before the patient come to be treated with bone marrow here, so 1 month before we treat them with fisetin. So like this, we eliminate the senescent cells after, before they do the bone marrow, aspirate, procedures and injection. So what we do after that is, we found, and I've published this, many years ago in Pittsburgh, we found that stem cells are very good, tools for regenerative medicine. The problem with stem cell, though, they respond to a lot of stimuli. So what we found here in 2003 and 2004, as you can see here, 2002, is we found that if you inject stem cells into a muscle and you injure the muscle, then the stem cells will repair the muscle. But what happened now, if you injure the muscle, wait 3 weeks, and after this, inject the stem cells. And what we found is if you inject the stem cells into a muscle that has been injured 3 weeks before, the stem cell becomes scar tissue. That stem cell builds fibrosis. So clearly here, we think that antifibrotic agent can improve the benefit of stem cells because a lot of patients that we see, they don't have an injury 1 hour before. Sometimes they have an injury probably 2 weeks before. And clearly, the micromilieu contain a lot of TGF-beta 1. Is it working in animal models? So this is the work we have done in Pittsburgh again. When I use stem cells alone here to improve muscle injuries, we improve the regenerative potential of those stem cells. But when I had losartan, then I further improve the regenerative potential of those stem cells by reducing scar tissues. I use here adult muscle stem cells. Some people here have used the same thing for adipose-derived stem cells. So clearly, we believe that losartan may improve the regenerative potential of bone marrow stem cells in the context of OA, because I guarantee you that in the joint fluid, you probably have TGF-beta 1 stimuli that will turn your stem cells into scar instead of having a benefit for those stem cells. So that's the second approach we're using here is after surgery, after injection of the bone marrow concentrate, we put the patient for 28 days on COSAR to prevent this fibrosis to develop after surgery. So this is our trial. So we do bone marrow injection to the patient, but 1 month before we start with fisetin to eliminate senescent cells. And 1 month after, we treat the patient with COSAR to reduce fibrosis. And here's a good example of a project that I said, I don't want to improve the surgery of the surgeon because those surgeons are very good to do surgery already. What I want to improve is the patient. I want to make sure the patient, is optimized for surgery by reducing inflammation and reducing scar tissue. So this is the trials here. And again, those patients, as you can see the design here, some patient got bone marrow aspirate only. Some patient got bone marrow plus fisetin, bone marrow plus fisetin and losartan, and bone marrow plus losartan. So clearly, and we believe that the addition of losartan and fisetin will further improve the benefit of bone marrow stem cell for OA patient. This trial started in 2020, but we recruit our first patient in 2021, and now we have over 30 patients that has been recruited. So again, I just want to finish here that, for me, the way I see orthobiologics in 2021, yes, you have PRP and bone marrow aspirate being your tool, your main tool to be used. But you have platelet-poor plasma now that people are working on, and you have adipose-derived stem cells, but also other stem cells that people are starting to use. But what I feel is those products is, it's good, but I really believe that optimizing the patient by using as an off-label either drug to eliminate senescent cells or drug to reduce, scar tissues, I believe that you will improve the benefit of the orthobiologics, in the future. And that's really what we want to do. So with that, I'm going to close here, and again, this is a team, the regenerative medicine team that we have. This is a team that we have here in Vail, Colorado. This is a team that we have in Fort Collins to do all the animal studies. The Proof Point Biologic team here is a team that do PRP and BMAC, all the time in patient, every day in clinic. Here's a team of surgeons that participate to those projects. And finally, this is the clinical fellows that work, with us to help us with this project. And I would like to thank you for your attention. Thank you very much.
Thank you, Johnny. Terrific overview. Great progress in not only your preclinical studies, but your clinical trials as well. We have first question from Dr. Guangzhou Zhai. Thank you very much for this excellent talk, Dr. Huard. Blocking TGF beta 1 with losartan is very interesting. I wonder if you looked at what happened to BMP pathway when you blocked TGF beta 1.
Yeah, this is a very, very good question, and we're still doing those studies. We do know that losartan is an angiotensin 2 receptor blocker that also block the TGF beta 1 pathways, but the downstream event is unclear now. We're doing some studies in animal study to try to decipher which pathways is being influenced. Also, I have to tell you that on the opposite side, losartan we know it can be used as a TGF beta 1 blocker, but losartan has a lot of off target. Losartan first, reduce blood pressure, as you know. So we think that losartan may do something else in addition to block the TGF beta 1. But clearly, we have done some study as well, by using, the nanofibers to deliver the TGF beta 1, blocker with Sam Stubb at Northwestern University. And we found the same thing. If you block TGF beta 1, you will basically improve cartilage repair. And I believe it's all about the dosing. I believe that in a neo-OA, you may have too much TGF beta 1. A small amount of TGF beta 1 it may be good to turn your stem cell to chondrocyte, but the huge amount probably lead to the deleterious effect. So I really believe that TGF beta 1, if you eliminate all the TGF beta 1, you're going to have an opposite problem, because I believe that TGF beta 1 is important for chondrogenesis and cartilage repair. But I think what losartan does is it reduces the excessive amount of TGF beta 1, maybe in the knee joint, and that is beneficial for cartilage repair.
Thank you. I was wondering a follow-up question on losartan. So the intra-articular injection for losartan, I was wondering if you could comment on what would be the uptake mechanism when you inject this intra-articular?
Yeah.
Is it cartilage your site of interest, or is it synovium? And how does the uptake mechanism look like?
Well, I think this is also a very good question here. Again, the main motivation to use intra-articular losartan was stemming from the fact that, if you give a blood pressure to your young athletes, you may drop their blood pressure. They may become hypotensive. Although when the FDA approved us to do this, we have only 2 patients out of like 350 patients that become hypotensive. So clearly, very few patients will become hypotensive. And this is why now we're not giving a 25 milligram capsule. We're breaking the capsules in half. We gave 12.5 milligram in the morning, 12.5 at night. And why I'm telling you all that is because the intra-articular was something that we explore. So first, I can tell you that the intra-articular losartan, I believe what we're looking at is cartilage. I'm not looking too much, at synovium and the effect on meniscus and ligament and a lot of things into the knee joint. I'm looking at cartilage. And I do know something is that, if the dose is too high, it's like those, anti-inflammatory drug, that people were injecting into the knee joint until some people find that they can be toxic to cartilage. So for us, our main readout was really, the type of cartilage you're making, and are we deleterious to the subchondral bone or, to the cartilage? And we found that we improve cartilage repair as well with losartan, but the dose here is very important. The dose, if it's too high, you're going to have a deleterious effect. The uptake mechanism, I don't know, because we always thought that losartan will block angiotensin 2 receptor blocker, to reduce the blood pressure. So, but clearly here, I don't know, I don't have all the science to analyze, what is the uptake, which cells is doing the uptake and everything. The only thing I do know, and this is under investigation as well, the only thing I do know that if you block the excess of TGF beta 1 into the knee joint after microfractures, you will have a benefit to articular cartilage, but too high as a dose is not good.
That's great, Johnny. Johnny, just as we're waiting for other questions to populate, maybe Rob can ask the follow-up question.
Sure. Okay. Yeah. Thanks, Johnny. That was great. I guess I was silent there. Okay. Here we go. My camera's off. A question about the selectivity of the fisetin pathway. I guess is the beta gal expression is the hallmark of the senescent cell population, probably the monocytes, you say. Is the mechanism of action of fisetin thought to involve beta gal itself, or is that just a secondary biomarker?
Yeah, I think that's a good question. This is a secondary biomarkers. Now we have also the UPAR, which is a marker that we have been using, to detect senescent cells as well. Now, if you listen to a lot of people around the country, now we have an arsenal of markers that's coming up to identify, senescent cells. And we do know that fisetin induce apoptosis, induce an apoptotic, cell death to those cells. We do know that, and the work with Jim Kirkland at Mayo Clinic has been very clear on that. The thing we don't know, though, is I don't think all senescent cells are the same. I really believe that you have the one that I talk about today, the one that are the bright senescent cells, bright on the facts, and the one that, fisetin seems to target, because fisetin seems to eliminate those cells. I really believe those cells that may have been in your body for 20, 30 years, because those cells do not die again.
Yeah.
But I believe you have an older population of senescent cells that are dim. They're not as bright by facts, when you stain with beta-gal. And the same thing is true when you stain with UPAR. Very few of those cells will be positive. They are very faintly positive, I would say. And I believe those cells may have been in your body maybe for 2 or 3 years. And the real question right now is, are those cells, gone, or can you reverse that? Would that be nice if I can take senescent cells and now have a drug to bring them back instead of killing them? I think the ones that are very bright, I don't think you can salvage them. They're probably very bad and you need to eliminate them. But the ones that are dim, I think it will be very important to see. So this is, one of the strategies that we're trying to look at is, can we develop new approach that will target the dim cells and trying to not only maybe eliminate some of them, but also trying to reverse senescence in some of them. That would be fantastic. So we're doing the work on this. We don't have all the answer for that.
I had a follow-up question on that, Johnny, about, I guess I had assumed that senescent cells really convey their clinical significance by the loss of reparative or anabolic activity. But the Jim Kirkland data was quite striking that they're actually, it's pro-inflammatory as much as loss of anabolic activity. Is that your thinking?
Yeah, that's my thinking. I mean, this is really an important point again. I think, again, I mean, I think what Jim, have shown, and they have different school of thought, because some people, do not all agree. Of course, we do science, and sometimes people agree and sometimes do not. But the bottom line, I really believe that those cells drive the disease. Yeah. I really believe that those cells, they are in your knee joint and they drive osteoarthritis. They drive, if they are, in your brain, we believe that they're going to drive neurodegeneration. And the most exciting part about this is, for many years for osteoarthritis, we're using anti-inflammatory drugs that we block the inflammation. But if you don't eliminate the cells that produce inflammation, then you know what, your treatment can be only, very good for a short time period, and after this, it come back. I believe, and now we're doing some work where we're using fisetin or any senolytic agent to eliminate senescent cells, and we compare this with, the regular anti-inflammatory drugs, and we want to see that, whether with the fisetin we're going to have a long-lasting effect on inflammation. Yeah. But with the NSAID and so forth, what you do is you reduce inflammation for a while, but those cells are still there. So I think they have 25 clinical trials around the country right now, and Mayo Clinic has probably 15 of them, for frailty, for neurodegeneration, for a lot of bone disease, and us, we're doing osteoarthritis, of course. And why they are so exciting is because we really believe that those cells are driving inflammation, and inflammation is driving aging. So clearly, this is very exciting.
That's great, Johnny. Johnny, so in your clinical trial, you're looking at senescent cells in blood. Now, what about local joint tissues and maybe synovial fluid? Yeah. probably you have the opportunity to look at synovial fluid and look at what happens in the local joint area. Would that actually help us understand more about these senescent cells, which are locally derived, as opposed to the one which are in blood? So just wanted to get your thoughts on that.
Well, I think that's a very good question, again here. First of all, I think the cells into the knee joint, the joint fluid, the problem with that technology is that a lot of people that come to see you doesn't have a huge inflammation flux. So the bottom line, you cannot aspirate a lot of things in a joint fluid. And what we do is we wash, we inject, saline, and we basically, wash the joint and try to aspirate that. But clearly, we have people that has a lot of infusion. If you bust your ECL, 24 hours later, you're gonna have a huge inflammation there. That's very easy to aspirate. But if you have osteoarthritis, you come to see a patient, sometimes you don't have a huge inflammation. So I think, I really believe that, those cells, what I would like to do is for any surgery, now it will be nice to do an aspiration to try to educate yourself, what am I dealing with here? I'm injecting something into a knee joint, but this knee joint here has, a lot of inflammation, or this knee joint has a lot of fibrosis, or this knee joint has too much angiogenesis. So clearly, I think that aspirating the knee joint, to get, a signature of what you're injecting to, it's important. And having senescent cells there, it's very new and novel for us. Are they the same as the peripheral blood and the bone marrow? Absolutely not. I don't think they are. And this is why we would like to do this SENNet proposal, and we would like to collaborate with you guys, is I would like to compare senescent cell from peripheral blood, bone marrow, and joint fluid. And I would like to see, what are they? And also I would like to do single cell seq to see, are we talking about the same cell population? But I can tell you already on the facts, they look totally different. And what they do in your knee joint, I'm sure they are not helping you. I'm sure that they can just, I used to say that they're playing against the surgeon. So every time you want to do a surgery, if you're dealing with this, they're going to make sure that they're going to, limit the benefit of your surgery. But how can you use this down the road is unclear, because I don't think that, you can aspirate, joint fluid in a lot of patients unless they have a lot of inflammation, and that makes the story a little bit more difficult.
Oh, absolutely. Rob, any last question?
Just one quick question, Johnny, about arthrofibrosis. I know we've seen some cases after arthroscopy that they're just very tough to treat. I wonder whether COSAR's got any traction there in those patients.
Wow, this is exactly, I think that's going to be the main target, because, after joint replacement, for example, very often, 25% of the patient come back because they have a very stiff joint.
Right.
And they have a lot of arthrofibrosis. So now we're trying to embark into this, what if you treat your patient with losartan? Can you reduce this arthrofibrosis? And it's a big problem, because the surgeon has to go in again and remove the scar tissues and everything. Yeah. So I believe that fibrosis, fibrosis and losartan is helping, microfracture right now. But I feel that, it will be even more important if you try to use it to eliminate or reduce arthrofibrosis. So I think that's something we're doing here in Vail, but I don't have all the results for that.
Thank you.
That's great, Johnny. We're literally at 8:59, and thank you so much for joining us today.
Thanks, Johnny. It was great.
Thank you, guys.
Have a good day.
Bye.
Thank you, everyone, for joining us today. We're going to meet on October 12th again for a lecture by Dr. Henning Galabay on early inflammatory arthritis. See you all on October 12th. And Johnny, thanks again.
Okay, thank you.
Bye-bye.
Bye.